Hello everybody. Part 1 of our lecture just ended. The most important point of the last part of that lecture was the product of one reaction is often the reactant for the next, forming a linear chain of reactions. So generally speaking, pathways do not stand alone, but they're interconnected with other pathways, and so it becomes essentially a very complex set of things happening inside the cell. I think that you'll understand better these metabolic pathways with an example. So the first one is a linear metabolic process, or a linear metabolic pathway. Now you notice at the bottom of the slide, we have a starting molecule A, then we have a molecule B, a C, and a D. You might remember from some basic chemistry that in a chemical equation, we have reactants, and then we have products. Now we know enzymes are the catalysts that speed up those reactions. So when we're thinking about the reactants and the products, let's take a look at this linear pathway. So that starting molecule will be our reactant of reaction 1, and it's going to make products, and those products essentially become the reactants in part B that are going to be going through that reaction of number 2. Reaction number 2 makes products we'll call C, and C now becomes the reactants for reaction 3. And then finally we get the final product of this pathway. But you can see that each reaction is making its own products, but the product becomes the reactant in the next step. So here we have a linear, step-by-step metabolic pathway that's very easy to see. Okay, so the second type of metabolic pathway that we're going to talk about in class, you can see here in the center, the cyclic type. So essentially, when you think about a cycle, I'm thinking about like recycling, upcycling, the idea that we are going through a set of processes and we are somewhere in there reusing some of the products, okay? And yes, the products can become reactants, but it feeds back on itself. So in the linear model we just showed, we ended with that product D, I think it was D, maybe it was E, but that very end product. And here, essentially, that very end product would feed back into the cycle, okay? And so this is really handy in biological systems because we're always going to have on hand all of the reactants and the products to make that cycle continue going. There's also a number of enzymes, okay? A number of enzymes that are going to be important to pushing the different steps along in the cycle. The branch type of metabolism we're not going to talk about. All right, when we think about energy in cells, I want you just to think about the energy currency of cells is called ATP. So again, we said it was a nucleic acid, adenosine, plus 3 phosphates. So it turns out that where the phosphate is joined onto that adenosine, each phosphate bond is what we call a high phosphate bond, high energy phosphate bond. So when you think about high energy phosphate bonds, it must mean that we have to add energy to get that phosphate bond to have the high energy to begin with. And then when we break it, we're liberating energy. So just be thinking, okay, ATP has 3 high energy phosphate bonds, and when we rip one off, we yield energy, right? And then if we want to take that molecule, which is now ADP, instead of triphosphates, it's a diphosphate, then we add phosphate back on it, we're going to have to put energy into it so that it is going to allow for that high energy phosphate bond to form between the second and the third phosphate. So we see that this is one of the chemicals, if you will, that's going to be changed and reverted back and changed and reverted back. We can keep on using this basic energy molecule. Okay, so ATP, if we go back to our beginning part of this lecture, is going to connect catabolism and anabolism. So let's think about it. I want to start with catabolism. Catabolism is one in which we are essentially breaking down complex molecules into simpler molecules. Where's my little thingies? There we go. All right, so if we take a look on the left-hand side, we see catabolism. So essentially we're putting in this case, it's going to be glucose, and glucose is going to get broken down and it's going to be fed directly into a series of steps or pathways that is going to eventually, you can see here, yield heat, okay? And we also see that this energy from breaking down this molecule is going to make that energy available to convert the diphosphate back into triphosphate. So catabolism is going to require energy. In your mind, you might know that for you to eat and digest a meal, you're burning a small amount of energy. So we're actually using some of the very energy that we need to ingest for our daily calorie count so that we can actually break down our food. So think about it that way. So catabolism breaks chemicals down, but it also requires energy, and that energy is going to go directly to ADP to make ATP. On the right-hand side, we're seeing that in this particular case, maybe the bacterium, I want to go back to bacteria, has to build up that lipopolysaccharide layer. So they're going to take glucose, which is part of that layer, and they're going to take some of that energy from that high energy phosphate bond, that third phosphate bond, and they're going to need it to put it into this reaction so they can build polysaccharides for the lipopolysaccharide layer. So anabolism, to build up things, we need to put energy in. So the way that we're putting energy in is we're stripping off a phosphate to have that high energy phosphate bond so that we can have the energy. It does get easier, I promise. Okay, so this is right off of your lab worksheet for the week. Draw a concept map illustrating the ways microorganisms perform carbohydrate catabolism and include the following words. So I want to show you what this looks like because I hear that students sometimes have difficulty with the concept maps. But essentially in carbohydrate catabolism, so essentially we're breaking down carbohydrates. When we're doing carbohydrate catabolism, and we're really thinking about glucose, okay? So glucose is the basic subunit of the macromolecules called carbohydrates. So it's going to be that source of energy that's going to go into the cells to eventually make the ATP, okay? So our cells and microbial cells can't directly use the glucose. They need the ATP. So we have to go through this catabolism events to do that. All right, so let's take a look at the concept map. So carbohydrate catabolism can occur in the presence of oxygen, and we're usually thinking a lot of oxygen. So for those of us animals are aerobic creatures, and certainly there are many microbes that prefer an aerobic environment. So if that's the case, then we're going to undergo cellular respiration, just straightforward, much like eukaryotic organisms. So it might be that we're trying to break down that glucose in a low oxygen environment. So low oxygen environment, we're thinking about anaerobic respiration here. No oxygen at all means we're going to go into this fermentation mode of catabolism. So essentially we're going to talk about it, cellular respiration, then we're going to talk about anaerobic respiration, and then fermentation. And you should know they all make itty-bitty amounts of energy, but cellular respiration makes oodles and goodles more than anaerobic and fermentation. Okay, for those of you that are still thinking about ATP, this is what it looks like. So here's our adenine with the ribose, so we call this adenosine. And here are our phosphate bonds, and you can see them here projecting out, kind of looking like they're really cool and groovy, but it's really this kind of, I think they call it a tilde, but this little sinusoidal snake-like looking structure. This is designating the high energy phosphate bond. So when we're ripping off the phosphate, we have to break that bond, and that's what's yielding the energy. That's also the bond that requires the energy when we go from ADP, adenosine diphosphate, back to the triphosphate. I don't think we need to stay on this. You can pause it if you want to read it. I think it's just a different way, a more complex way of the things we've been saying. And I'm not talking about reducing power. So you can skip that. Okay, so we have the three main carbohydrate catabolic pathways here. So what I want you to see, aerobic, on the left, anaerobic in the middle, and on the right-hand side, fermentation. And we see that all of these pathways are going to yield a small amount of ATP right at the beginning. I also want you to see that this process here, it looks like a prong, or a tuning fork. This is what we call glycolysis or glycolysis. Okay, and it is the same for aerobic, anaerobic, and fermentation. Essentially, it starts exactly the same. So you see some other things, just kind of look at the picture and go, yes, okay, I see they're exactly the same. And it says on the left-hand side, it yields 2 ATPs. So that is the same. Now I want you to jump down to the bottom, the maximum net yield. So with our aerobic respirators, the ones that absolutely require oxygen, we know that they're going to make somewhere between 36 and 38 molecules of ATP. Okay, that's what's going to happen as a result of this this multi-step process here. Okay, so we're going to call this cellular respiration, and it's going to go through these steps. The one in the middle, anaerobic, they usually are the presence of some amount of oxygen, but the thing is they're going to end up making different products. So we didn't look at this with the first one, but I want you to look at this with the second one. So they are using a non-oxygen compound as the last electron acceptor. So essentially what we see with this particular type of carbohydrate catabolism is that it is going to make far less ATP. It says 2 to 36, but 36 would be on the high side of what most anaerobes could do. So it's usually much less than that. But you can see at the bottom of this, you see that ATP looks like a little sun showing us, woohoo, energy has been made. Now, for fun, let's go back to aerobic respiration, and you see that, yes, indeed, oxygen is the final electron acceptor. So this is super important to making this net yield of 36 to 38 ATPs. The last pathway, fermentation, you can see it also starts with glycolysis. Looks exactly like the other two pathways, but now we skip the Krebs cycle, and we're also going to skip this part here. It's actually showing us a membrane. It says the electron transport system. So in fermentation, there is no oxygen, and essentially we have no Krebs cycle or electron transport chain, and instead we are going to use organic compounds as the electron acceptor. So what this is either alcohol, ethanol, things like acids like lactic acid. So when you're working out and your muscles are depleted of oxygen, then fermentation essentially is happening in your muscles, and you get the buildup of lactic acid, which then gives you sore muscles. So one thing you might do after a workout is drink a lot of water, get a massage, or kind of just, self-massage or hammer your muscles so you can get some of that lactic acid to release. But you see the end product is only 2 ATPs. We have one more video, and we're at 13 minutes now, so I'm going to end this here, and then I'm going to talk about what's happening in glycolysis in the Krebs cycle and the electron transport chain. There's not a lot of that on the study guide or the worksheet for the week, but I do want to make sure that you've got some words and slides to go along with those steps of the pathway, so you feel a little bit more comfortable with the language. Okay, I'll see you next time.